Hello, everyone. This is Pocket Size Bio, Dr. Casco. I'm back with another video. This is chapter 1.3 or part 3 um, of the video series that I'm making. And this particular part is eukaryotes and the origin of the eukaryotic cell. And along with this part, I'm adding the module on model organisms, right? So this video might be a little bit longer, or maybe twice as long, sorry, as the previous ones. So let's get to it. All right. So a little bit of introduction here. So we're going to talk about eukaryotic cells, more specifically with some emphasis on prokaryotic cells. But the main scope will be eukaryotic cells, right? And we know maybe through high school or undergraduate biology, eukaryotic cells in general, right? They're a lot bigger than prokaryotic cells. They have a much larger genome size, right? Variation in chromosome number. There's a lot of variation in cell morphology, right, in their structure, therefore their function there. Eukaryotic cells, as you can see here, in this particular diagram, they contain a lot of structures within the body, right? It's not as simple as the prokaryotic cell. You can see there's a lot of organelles, right? Or little substructures, little mini organs that are, well, a lot of them are kind of membrane enclosed, right? Where they're performing their own chemical processes, biochemical processes and reactions. And right? Among those, you have areas where you just have little macromolecular assemblies, right? Aggregates of chemicals that are creating their own, um, molecular machinery without necessarily having a membrane, right? So eukaryotic cells can live independently as a single cell, or they can be multicellular, right? As we know. Um, so we're going to get into some characteristics, right, about eukaryotic cells and a little bit about how the eukaryotic cell came about. So, eukaryotic cells contain a variety of organelles. I'm going to briefly go over these, right? In the later chapters, of course, we get into them in much, much more detail. All right. And I'm going to be going to this diagram as we go along for reference, right? So by definition, right, an eukaryotic cell keeps almost all their DNA, right, inside a membrane enclosed internal compartment, which is the nucleus, right? And as you take a look at this picture, generally the nucleus is the largest organelle, right? And it houses the DNA, right? And it's how the DNA is packaged, right, and coiled is largely due, to proteins called histones, right? And there's discrete amount of protein packages that we know them as chromosomes, right? And different organisms could have different numbers of chromosomes, right? But more on that later in the later chapters. Ultimately, you have this structure. Let me zoom into that, right? So you have this structure that is enclosed in a membrane. It's a double membrane, right? And referred to specifically as the nuclear membrane, right? And it has little pores, okay, all throughout this kind of spherical structure. Of course, you're seeing a little cross section here, for example, of the nucleus, and you essentially, you have this little nuclear envelope, and it has these little pores in which material can come in and out, right? Um, inside the nucleus and outside into the material could come into the nucleus and or outside, right, into the cytoplasm, which is basically the fluid, right, in which everything gets suspended in the cell. More on that later. So eukaryotic cells are generally, 10 to 30 times bigger than bacterial cells or prokaryotic cells, right? And in terms of volume, they could be 1,000 to 10,000 times larger than prokaryotic cells. So in general, they're much, much larger, right? As you see in this figure here, you see a lot of filaments, right? For example, these little green filamentous material here. You see some blue little filaments, little lines there. You see some red little little lines there, right? Let me just zoom out. So all of these are essentially, the cytoskeleton, right? As I go along here in the notes, and there's many types of cytoskeleton fibers. You have microtubules, you have little actin filaments, right? You have intermediate filaments, and they have a wide variety of functions, right? But they help kind of keep the cell, right, have a certain shape. They also enable the cell to move around through a particular area. They help areas of the cell extend, basically to create a process called phagocytosis, right? Which you see here, part of the membrane, right, of this particular cell that you see here, which is a phagocytic white blood cell, extends, right? And essentially will engulf some foreign invader, in this case, a bacterial cell, right? You see these protrusions coming out, right? All of this is happening in part by this cytoskeleton, right? So definitely there's a lot of functions involved in this whole cytoskeleton, right? Um, Definitely helps in the movement of organelles, right? Large molecules and a lot of molecular machinery there. Okay. In terms of cell division that we're going to take a look at, they help in chromosome separation, right? During um, movement of chromosomes during metaphase and anaphase and so forth, right? So they have a very important role in the cell regulation, right? So a cytoskeleton, and then you have some organelles that are membrane enclosed, right? So they have a cell membrane surrounding um, these organelles, right? Which include the endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, right? And several of these you can see. Let me just zoom back in here. Okay. Here in white, you have the endoplasmic reticulum here. All right. Um, this vast network of flattened sacs and tubules, right? You have the Golgi apparatus that seems here, all with its little border of cell membranes. Okay. You have little peroxisomes here and so forth, right? So each of these organelles, of course, have their own particular function, right? Endoplasmic reticulum has a single membrane. My apology if I said double membrane. Um, uh, is the most extensive organelle, right? And it's quite interesting. It's one of the main functions that it does is to create more membrane, right? Or the membrane components are kind of manufactured here, right? And it helps begin this whole kind of transport system or endomembrane, transport system, right? Where you're starting to secrete material, right? You have the Golgi apparatus that you can see in this area here that also is a single membrane that kind of receives the packages coming from the ER and it kind of tags them, modifies them, and packages material, let's say proteins for transportation, ready for secretion internally within the cell or even extracellularly outside of the cell, right? Golgi apparatus. You have little lysosomes there. That these are small, irregularly shaped organelles, right? That primarily serve for intercellular digestion. So you essentially have, um, I apologize for the little ring there. Um, Intracellular digestion, where you have um, these lysosomes engulfing little foreign particles to be broken down and whatnot. Okay, you have peroxisomes. These are a specialized type of vesicle, right? Little kind of membrane encapsulated little ship, if I can call it, right? Which are essentially contain H2O2 that help inactivate toxic materials or toxic molecules, right? Like oxygen reactive species, right? And these vesicles, right, they do help move material in and out of the cell through two particular processes called endocytosis and exocytosis, right? That you can see here, where the membrane, of course, it's all the same membrane that helps encapsulate things. Like the cell itself has a plasma membrane, but all these little ships, all these little vesicles, the Golgi, are all made up of the same thing. So when you want to bring a large amount of material in, or large particles in, the cell membrane kind of bends inwards, or creates a little inward folding, and it creates a little vesicle, right? A little sphere containing the material that you want to bring in, right? That process that we'll see later on is called endocytosis, basically importing material. And it'll go to wherever it needs to go, right? At the same time, if you want to expel material outside of the cell, you essentially will bud out a little vesicle to move to the cell membrane and export that material outside. So they're both kind of reverse processes, right? To ultimately to move material. Okay. Ultimately to move material. I'm going to pause the recording in a bit. Okay. Sorry about that. So other double membrane enclosed organelles, right? So these are organelles that contain two layers of cell membrane, right? Which is the mitochondria and chloroplast. Okay. So I'm going to go back to my diagram here. All right. And if you see here, you have the mitochondria, and this particular cell seems like it doesn't have chloroplast. All right. So what you need for this particular organelles, right? These two particular organelles, right? You have mitochondria and chloroplast, and mitochondria, one of the um, one of the organelles, if I can say, that provides for the theory on how the eukaryotic cells came about, which I'll talk about later. Mitochondria is essentially for performing cell respiration, in which the cell takes in oxygen and essentially harnesses energy, by oxidizing food molecules, for example, sugars or fats, to produce ultimately ATP, right? So you want to produce energy in the form of ATP to do your other cellular processes, or many cellular processes, right? Um, so via oxidation reactions, right? As opposed to chloroplast, which essentially performs photosynthesis, and any any organism or cell that performs photosynthesis is essentially um, an autotroph, right? That will produce food, right? Basically, glucose. All right. Some example of cells would be plant cells or algae, right? And the whole purpose of this process is essentially to obtain energy from sunlight, right? To synthesize carbohydrates using very simple materials like carbon dioxide and oxygen, and water, right? H2O there. Um, we all, I'm sure, we're all familiar with this process. Um, and Interestingly, right, generally, half of the cytoplasm is occupied by membrane enclosed organelles, right? Like the ones I mentioned before, right? And then the fluid surrounding those organelles, right? Basically, this area in gray that you see there is referred to as the cytosol, right? And within swimming in that cytosol, you have ribosomes, right? One of the most important organelles, essentially. Um, that produces protein, right? Manufactures proteins. But aside from that, within the cytosol, within this solution, right? Um, most metabolic reactions are taking place. Okay. As I mentioned before in the beginning, some organelles lack a membrane, right? So really, they're just a bunch of an aggregate, right, of different proteins, molecules that are working together in a particular area, right? Um, which are generally called biomolecular condensates, right? And an example of that would be the nucleolus, right? That you can see here. Not enclosed, but you have this area where you have specific molecules, right, in close proximity performing chemical reactions, right, for the nucleus and for the cell. Okay. All right. So talking about mitochondria, right? Mitochondria evolved, right, from a symbiotic bacterium captured by an ancient archaean prokaryote, right? Or archaea. So let me just go a little bit down here so you can see. Before I get into that, just a very nice representation, a simplified diagram of what I just mentioned, right, of the cell. You have your little organelles that are colored in, that are membrane bound, right, that I just previously discussed. Okay. Now, I'm going to zoom in a little bit here. All right. So, uh, the emergence, this is quite interesting. The emergence of the first eukaryotic cells, does represent a fundamental question, right? In evolutionary biology, in general, in biology itself, in particular cell biology, right? How did the first eukaryotic cell come about, right? So evidence does suggest that the first eukaryotic cell, originated around 2 billion years, right? 2 billion years ago. And by a process essentially that it wraps around the idea about a symbiotic merge, right, between an archaean prokaryotic cell and uh, a bacteria cell, right? And for the first couple of billion years, right, if I can say that, because it's such a long time, the world was pretty much only prokaryotes, right, for over 1.5 billion years. And um, around that time, you have cells that look like mitochondria, right? You have cells that were very simple and primitive and small and had particular functions, simple functions, right? And the reason why is that scientists, you know, see that um, the eukaryotic cell came from this kind of endosymbiotic theory is because mitochondria does resemble some similarities with prokaryotic cells, right? They're very similar in size to bacteria, right? They possess, and this goes with chloroplast, by the way, right? That we're going to take a look. Mitochondria has its own DNA, right? So aside from the nucleus that an eukaryotic cell has, mitochondria and chloroplasts actually have their own DNA, right? In little circular kind of plasmid-like, right? And they resemble bacterial genes, right? And mitochondria replicate by division, right? Little mitochondrial division. And they can replicate independently of the cell replicating, right? So these bacteria-like features, right, gave scientists insights onto this kind of evolutionary origin of eukaryotic cells, right? And these type of, bacteria-like structures, um, essentially, right, suggest an evolutionary origin, right, from aerobic bacteria that were actually captured by anaerobic cells. So it mentions aerobic bacteria because mitochondria need oxygen, right, to function, right? So there must have been some relationship between anaerobic cells and aerobic cells at that time, right? Because we're going to take a look at more in detail now. So, um, uh, the endosymbiotic relationship, basically mitochondria, um, likely evolved, right, from aerobic bacteria that was captured by anaerobic cells, right? Anaerobic cells meaning that they don't use oxygen in their metabolism, right? And that led to an endosymbiotic relationship. Okay, so I'm going to go a little bit down, a little bit up here. So you can see on these panels. And right? This endosymbiotic relationship. And essentially, this relationship, right, enabled a mutual metabolic support between each other, right? Um, where you have this primitive mitochondria that is producing, these food molecules from oxygen, right? That could be used as um, a source of food for this anaerobic life form, right? Archaea being a lot bigger, right? A little bit bigger, could serve as that host or as that medium to thrive in, right? So you have this archaeal host, right? And that bacterial symbiote that they kind of share their 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 structure, their bodies, their cytoplasm, right? So, um, in terms of that archaeal host, right? The archaeal origins of eukaryotic cells, right? Archaea are believed to be that ancestral capturing cell, right? And reasons for that is because of genetic and molecular similarities to actual present day eukaryotic cells, right? And of the particular lineages, or lineage of Archaea is the Asgard lineage of Archaea, right? They're the most similar in terms of DNA sequencing due to samples on seabeds, right? That have sequences similar to eukaryotic DNA, right? So, of course, scientists, collected these samples. They made cultures of these samples and sequenced um, to get some idea on similarity, right? Um, that study or studying this archaeon basically took 12 years, right? And they're very unique, right? In their um, um, in their structure, right? In their DNA sequence, right? And you can see an example of a scanning electron micrograph of an archaeon, right? Or an Asgard archaeon in culture, right? So there were very interesting, you know, physiology going on there. You had this archaeon body, and you had these little filamentous protrusions there that were um, that were being shown, right? So, some features that could be seen in these particular structures, right, is that it does show some initial ectosymbiotic relationship with other bacteria and archaea, right? Um, that type of relationship where you have these kind of ectosymbionts and you have these protrusions that are connecting to these could have led to these to the engulfment, right, um of one to another, bringing it inside its body, right? Um, one way or another, right? Essentially kind of undergoing phagocytosis, but not digesting kind of situation there, right? Um, so this theory kind of led to the idea on potential mechanisms, right? On how this Asgard Archaeon brought in these aerobic, prokaryotic cells, kind of starting the eukaryotic lineage, right? So an example of what I'm referring to is here, right? Ultimately, you have this um, archaean, a lot larger archaean. You have this bacterial cell with bacterial DNA here that is being engulfed into the body of the archaean, but not digested. Eventually, all these protrusions and filaments kind of widened and kind of formulated a cell structure. And these little gaps were a kind of proto form of certain organelles, right? Like the endoplasmic reticulum, nuclear envelope, and so forth, right? Um, In terms of chloroplast, right? Because initially you had mitochondria first. Um, Chloroplast evolved from that symbiotic photosynthetic bacteria engulfed by an ancient prokaryotic cell, right? So, um, as we know, photosynthesis is occurring in chloroplasts, right, in plants and algae, and they essentially use sunlight energy to synthesize carbohydrates, right? And chloroplasts also have a double membrane, right, similar to mitochondria. They show, circular genomes that within their structure, and also they reproduce through division, right? Let me go a little bit higher here. Here's an example of a chloroplast organelle, right? And again, so something similar must have happened where you have this symbiotic photosynthetic bacteria that was captured in and retrieved into the body of of an ancient eukaryotic cell that already had mitochondria inside it, right? So they engulfed mitochondria, and maybe some of these cells in a population also engulfed, these photosynthetic prokaryotic cells, and essentially led to this, you know, um, uh, photosynthetic eukaryotic cell, right? Again, maybe similar process by phagocytosis, right? And it's quite interesting because some of the ideas going on is that, you know, um, having chloroplasts could have relieved, right, these heterotrophic cells to constantly need to go searching for food, right? Searching for prey and hunting and so forth. So instead of doing that, they have, their own source of energy, right, of food production, um, which ultimately led to these modern autotrophic cells, right? Um, yeah, so it's, some kind of, if I can say, some perks, right, of being equipped with chloroplast. They don't need to chase other cells for nourishment. Oh, my apologies, right? Um, they don't necessarily need, and due to that, they don't need to necessarily change their shape all the time to go phagocytize, if I can say, other cells, right? Instead, they could just have a much sturdier formation, right? Maybe the formation of cell walls and even protecting itself from other predators, right? Single-celled predators. So it's quite interesting how that could have came about, right? Um, so instead of having that hunter-gathering style, they adapted this kind of farming strategy style, right? Where they're producing food molecules, having protection, having stability, not necessarily a need to be moving around, right? Okay. Let me just go down a little bit. Fungal cells is also interesting there. They represent a different type of lineage, if I can say, or a lifestyle that's different from strict heterotrophs or strict autotrophs, right? They contain mitochondria, but they don't have chloroplasts, so they can't photosynthesize, right? They do have a cell wall, different from a cellulose cell wall. They have a chitin cell wall, right? But it is a tough outer wall. It does limit rapid movement and cell engulfment, but um, instead of constantly hunting, they're basically scavenging, right? They're feeding off nutrients of um, organisms that have, died, and they're secreting molecules that could be taken into later advantage of these scavengers, right? Of these fungal cells. So, as we all know, modern or fungal cells that we think about, they perform extracellular digestion by secreting digestive enzymes, right? To break down basically dead material, right? Organic matter. So that kind of reflects that idea of scavenging behavior, right? Ultimately to um, extract, nutrients, right? Okay. So now that we kind of get a sense of how these cells came about, how the eukaryotic cells came about, right? Um, Eukaryotes, you know, have hybrid genomes, right? And again, understanding the endosymbiotic theory, you have this concept, right? You have most of genetic material stored in the nucleus, but you also have small portions of DNA, even within our own bodies, right, that are remaining in organelles like the mitochondria and the chloroplast, right? So hence the idea of hybrid genomes. Okay. And definitely the DNA that you see nowadays is not like in the past, the reduced versions of um, the corresponding bacterial genomes of these ancestral mitochondria or ancestral chloroplast um, uh, cells, right? So if you were to take a look at some of the specifics within a human cell, for example, the mitochondrial genome comprises only roughly 16,000 nucleotide pairs, right? Which would code for 13 proteins and a set of 24 RNAs that would be involved in protein synthesis of just mitochondrial DNA, right? Or mitochondrial genome. So um, uh, it doesn't necessarily mean that they've completely disappeared, but um, it could have integrated inside um, the host cell nucleus, right, into that larger nuclear genome, right? So nuclear DNA of animals, does contain genes, right, that would code for proteins that would be essential, right, for the mitochondria, right? And respectively for plants and algae in the case of chloroplast, right? So that's good, clear evidence, right, of some bacterial origin, right, or this endosymbiotic theory that would show some form of evolutionary history, you know, um, in terms of the rise of eukaryotic cells, the process of endosymbiosis, and that concept of gene transfer, right, from mitochondrial genomes integrating itself into larger nuclear DNA genomes, right? So now we're going to get into a little bit of about eukaryotic genomes in general, right? I'm going to go a little bit higher here so you guys can see. All right. So, quite plainly, eukaryotic genomes are big, right? Um, and of course, if we take a look over time, we have been talking about how change over time, um, in the term of symbiosis, right? Natural selection has favored mitochondria, with small genomes over time. You want to just have the clear and concrete essential information, right? Um, Nuclear genomes of most eukaryotic cells have tended to be enlarged, right, or large. Um, and reasons for that, right, could be um, could be due to larger cell size in eukaryotic lifestyles, right? So having larger genomes, it may offer advantages, right, to predatory cells, okay? Which generally means that they need to be larger than their prey, okay? So, um, yeah, so eukaryotic genomes have become 100 times larger than bacteria and archaea cells, um, integrating their genomes within the larger nuclear, um, genome pool, right? Genetic material, if I can say, right? Let me just scroll a little bit higher here. So in general, right, eukaryotes not only possess more genes, but also have significantly more, non-coding DNA, right, compared to prokaryotes, right? Um, we're going to look into this a lot further in a lot more detail in the later chapters, right? Non-coding DNA that doesn't necessarily produce or translate to create a protein, right? Um, so they do have a lot more, right? In the case of the human genome, you have approximately 700 times more nucleotide pairs than the genome of E. coli, right? but only about 4 times as many protein coding genes, right? Not as many. So that's quite interesting, right? Um, Around 98.5% of the human genome, right, doesn't code for proteins compared to 11% in E. coli, right? It doesn't code for proteins. So a lot of our genome really doesn't code for anything. It was often referred to in the past as junk DNA, but definitely there's a lot more insight nowadays, right? Regulatory elements and such. Okay. So let me go a little bit here. So table 1.2, as you see here, compares the estimated size, right? The genome sizes and genomes numbers, right? For these particular eukaryotes, right? Comparing also with E. coli, right? So you can see some significant difference between complexity and non-coding DNA um, content. So approximate size of the genome and approximate number of just protein coding genes, right? Um, Kind of fortifying what I'm mentioning, right? About non-coding and percent of non-coding um, DNA. All right. So before I move on to the next section, I just want to kind of show here, right, that let me just move this here, make this kind of half-half. Sorry about that. All right. So this little diagram is essentially a model of how uh this idea of right of evolution of eukaryotic cells came about, um, where you have this endosymbiotic theory, right? Which would merge kind of this um, mitochondria to produce a single-celled eukaryote over time. And then that single-celled eukaryote that integrated mitochondria into its body, furthermore integrated chloroplast, ultimately, which led to plants, those that couldn't um, take in, right? Chloroplast ended up turning into animals and fungi, right? So it's a very nice depiction of what I was referring to, right? Um, Figure 130 is basically genome sizes compared, right? Where um, you would essentially have, nucleotide pairs between different species there. And as you see in terms of simplicity, you have bacteria and archaea and protozoans, um, smaller, gene sizes or genome sizes. Um, oops. Of course, protozoans being so diverse, their genome size spans greatly there, right? But there is a trend where you have, as you kind of go along in a evolutionary tree in complexity, genome sizes do tend to increase, right? From prokaryote to eukaryotes and even the different groups of life, right? Okay. Let's uh continue on here. Okay. Just zoom into this photo. All right. So eukaryotic genomes are rich in regulatory DNA. Um, so certain kind of concepts that we need to go over, you should know. Definitely, we get into this in a lot more detail. Non-protein coding DNA, right? And again, I just mentioned this, right? It's referred to as was referred to as junk DNA, right? And it has been retained through evolution, right? The book mentions it's akin to maintaining an archive of old papers, and as it's easier to keep everything rather than kind of remove waste energy and effort, right? To be removing what's not necessary, it's better to just keep it, right? Um, and just accumulate and accumulate. Um, and That's not always the case. There are there are some eukaryotic species like the pufferfish that they've managed to eliminate, right, large amounts of non-protein coding DNA with no adverse effects, right, on their physiology, on their anatomy, on the structure and behavior um, of of their genome relative to other organisms with a much larger genome. So it's quite interesting how you have a species um, organisms that are able to do that, right, while others didn't, right? So in terms of genome structure and function, right? Despite whether you have a lot of junk DNA or even in nicely compacted, useful information, information rich coding DNA like the pufferfish, right? Um, non-protein coding DNA would always be a lot more than protein coding DNA, right? As I mentioned before, junk DNA scientists have learned and are still discovering, right? There's a lot of regulatory elements embedded within these junk DNA sequences that regulate gene expression, right? And I apologize for my video that I kind of got stuck, so I deactivated there. Sorry. Um, so regulatory DNA controls when and where, right, genes are activated. So there's a lot of mechanisms for gene regulation, right? Not just general expression, but also kind of turning off and on genes, right? And that's very, very important, right? As we're going to see, because it ultimately helps for development of certain structures. It helps develop the diversity of cell types that we think about, right? And a lot of specialized functions and so forth, right? Okay. Eukaryotic genomes defined the program of multicellular development. Um, So cell diversity, right, in multicellular organisms, as I kind of just mentioned in terms of gene regulation, right? Even cells within a single organism and plant have enormous amount of diversity, right? We all know that there are many different types of cells, like blood cells, skin cells, bone cells, nerve cells, which are very, very dissimilar, right? And here's an example of just one diverse amount of cell, size and shape. You have a neuron, highly ramified structure, right? Um, you have the axon and the dendrites, that neuron body, compared to another cell of the same individual, basically a human, a neutrophil, right? Part of the immune system, right? But um, and they're so different from each other. So there's a lot of diversity in cell size and shape due to these mechanisms of regulation, right? Um, what is so interesting, right, is that despite diversity, all cell types, right, they trace back to their origins to a single fertilized egg, right? To that zygote that we think about, that single cell where we come from, essentially, right? And which contains identical copies of the species genome, right? So developmental regulation, right, of gene expression, as I mentioned before, right? Um, the reason why there's cell differences is because you have selective utilization of genetic instructions, right? Um, developmental history, environmental cues, as the embryo, as that single cell is growing after fertilization, during embryonic development, you're having certain switches turn off and on that is leading to um, uh, different pattern formation, right? So definitely DNA does not function as just as a 0 100 blueprint fixed, right, for every single cell, but it's um, basically portrayed for cells that they act as multi-purpose machines, right? And they are capable of responding to environmental signals, right? That can activate certain genes accordingly to whatever the needs you have to develop, right? So, um, yeah, role of regulatory proteins and non-coding DNA. So a little bit on how this works, right? You have these regulatory proteins and these non-coding RNAs. Sorry. Um, so many genes in eukaryotic genomes, they encode proteins that regulate, right, the activity of other genes primarily through transcriptional regulation, right? And there's many transcription regulators that would bind to specific DNA sequences and would essentially give the okay or not to control gene expression levels and patterns, right? Um, and so forth. So eukaryotic genomes, right? They produce, aside from these regulators, these transcription regulators, they also produce non-coding RNA molecules that could also, influence gene expression. There are many diverse mechanisms, right? Even though the RNA molecule itself is not translated into a protein, right? So there's different ways in which you can regulate, okay, gene expression. One thing that's interesting that the book kind of mentions is that, genome as hardware and software, right? The the eukaryotic genome, right? Not only, cellular hardware, but also stores the software that governs how the hardware is utilized. And I kind of mentioned this in the first section, right? How the information in the DNA produces the protein, right, via transcription and translation, would ultimately comes back to kind of regulate the software, right? So this software includes, regulatory proteins and non-coding RNA that control gene expression, right? Ultimately controlling gene functions and so forth. Okay. Um, as we get into the actual book, and we'll see more in detail how, cells, they signal each other, neighboring cells, and they coordinate with neighboring cells to ultimately make a very intricate system, right, of one's own cell behavior, right? That is kind of communicating, right? So there's a very heavy element of control and synchrony between many cells that ultimately drives development and internal control mechanisms, right? Um, Essentially to control cellular activities, right? Um, I like to believe it's very heavy on signal changes and exchanges, right? Stimuli making cells kind of react in a particular way to drive a particular function, right? Or process or end goal, if I can say, right? I'm gonna go a little bit higher. Okay. So let me just kind of, all right. So an example of genetic control, you have um, this concept of genetic control in multicellular development. There's an example of the Snapdragon um, anti-renum, renum. Um, and you have a mutation then on one of the flowers, right? If you see here, you have flower formation, right? But if you have a mutation on a single gene that causes a mutation for a single gene, right, that is coding for a regulatory protein, it would cause, instead of flower development, you would have this kind of leafy shoots growing, right? So definitely gene, of course, regulation can influence the idea of formation and structure, right? Based on what is being turned on and turned off, right? Okay, just a moment here. All right, sorry about that. Okay, so continuing on, many eukaryotes, right, live as solitary cells, and many species we that we know of. You can see some examples here, right? They live solitary lives, and they exhibit many diverse or highly diverse lifestyle, right? Hunting, photosynthesis, scavenging, and you even within these eukaryotic structures or cells, right? There's a lot of diversity, anatomical variety, right? Um, and you can see some of these single cell eukaryotes, and you see such diverse structures, right? You can have sensory structures like little yeah, fine hairs to detect um, um, a type of stimulus, a change in wave in the water, wave motion, right? Or picking up a particular molecule, photoreceptors, right? Cilia or appendages for locomotion, um, particular parts to break down food or capture food, right? Attacking prey or attacking predators, and so forth, right? Life has developed so much diversity in terms of structure and function. It's quite fascinating, right? And these are electron micrographs that you can see of some examples of single cell eukaryotes, right? Of protist, for example. Um, Here you have a heterotrophic protist, right? Dedenium, um, belonging to a group of ciliates where they have these little hairs, right? Coming in and engulfing a another cell right there. Okay. So yes, intricacies and complexities of single cell eukaryotes, right? Um, there's a lot of versatility we're going to take a look at, right? And there's a lot of complexity in the behavior, right? Only because they're small and generally simpler doesn't necessarily mean that they're have simple behavior there, right? Um, and yeah, especially when it comes to proteins, right? There's a lot of variety, and there's a lot of versatility when it comes to structure, right? And function. Okay. So this last little part um for this particular section, I may create an extended video for the model organisms. Um, so it'll be technically 3B. This would be 3A, this video. So overlooking single cell eukaryotes. So humans um, often focus on plants and animals, right? Overlooking single celled organisms, right? Because of course they're just microscopic. It's quite difficult to see on a daily basis there. Um, but DNA comparisons have shown, right, that the genomes of single cell eukaryotes, they show more diversity than those of multicellular animals and plants, right? And if you were to see this in this kind of tree of life, right, take a look at all this diversity of a single-celled organisms, right? There's so many different branches, amoebozoa, right? You have the foraminiferans, you have the diatoms and the oomycetes, you have the ciliates, right? You have some predictive haplotypes with predictive lineages, um, uh, algae, right? Mosses and flowering plants, extending right into multicellularity. You have euglena, fungi, but if you take a look, right, very smallly branching out, flowering plants, fungi, animals, right? So they're quite diverse, right? If you were to position them. So genome data, right, has enabled scientists to place, um, very single-celled eukaryotes, right, in the tree of life, uh, quite accurately, right, due to DNA sequencing. So this also helps in investigating the origin of multicellularity, right? We kind of figured out or have a theory on how the eukaryotic cell came about, but what about multicellular organisms and how they came about, right? Of course, there are some methods that scientists already have established there on multicellularity, right? Um, so, by having such a massive amount of genomic data, from single-celled organisms, from multicellular organisms, right? You have scientists exploring the origins of multicellularity, ultimately to give that to gain more insight on evolution, right? And that evolutionary trajectory on complex life forms, right? So to kind of conclude this little section, and I apologize for the bell. The study of these organisms, right, on single-celled eukaryotes does provide valuable clues on the evolutionary past of all life forms, including humans.